And get started. Welcome everybody to today's webinar on the role of Epstein-Barr virus in multiple sclerosis. We're honored to have these two distinguished experts with us today, and we're going to delve into the fascinating topic that's quite clinically significant. First, I want to introduce Marianna Cortese, a senior research scientist at Harvard School of Public Health. Dr. Cortese's expertise lies in identifying risk factors and biomarkers for multiple sclerosis, as well as other neurodegenerative diseases. And she brings a lot of valuable insights that I'm excited, as I'm sure all of you are, to hear on the complex interplay between viral infections and neurological disorders. As well, we have Dr. Kjetil Bjornevik, an assistant professor of nutrition and epidemiology. Please feel free to correct me if I butchered either of your names. He also brings a lot of experience with new biomarkers, risk factors, and treatments for neurological diseases, including multiple sclerosis. And his clinical research adds a unique perspective to our discussion today. And together, they're going to shed light on the recent advancements in understanding how Epstein-Barr virus is influencing the development and progression of multiple sclerosis. So without further ado, let's delve into this compelling topic and gain some valuable insights from our esteemed speakers.
Thank you so much, Maggie. So let me try to share my screen. Can you see and hear me well?
Yes.
Perfect. Okay, so let's get started. Okay, so thank you so much for the invitation. We're very happy to be here to talk about Epstein-Barr virus and MS. Okay. So in this talk, we're going to give a very short introduction on MS as a disease and talk a bit about the role of infections in MS, specifically focusing on infectious mononucleosis. We're going to go over our recent publication on the role of the Epstein-Barr virus in MS etiology, and then in the end discuss some various aspects and implications of this paper. So you may all be familiar with MS as a disease, but just to shortly summarize, it's a disease that's quite different from other neurological diseases because it affects young adults. And this means that those affected by the disease, they live with the disease for many years, maybe 30, 40, even 50 years. On this picture, you can see a cartoon that nicely illustrates the underlying pathology of the disease. So on the left side, you can see an image of an actively demyelinating MS lesion stained for myelin protein in red and neurofilaments, which are particularly abundant in axons in green. The arrow shows area of demyelination, and that is the part of the axon that has lost the myelin sheet. And loss of this sheet makes the neuron vulnerable to damage and can cause them to die, thereby causing neurodegeneration, a permanent loss of neurons. So the cause of MS has been a topic of much discussion and disagreement ever since the disease was first defined more than 150 years ago. And still today, when you read articles about MS, you will usually be met by a description similar to this, that it's a disease of unknown etiology, but that environmental, lifestyle, and genetic factors likely play a role. With this understanding, it's difficult to find interventions that can help to reduce the risk and prevent the disease. So in our study, we aim to provide a better answer to this question, namely what is causing MS. So it's interesting when you look back in the literature that the hypothesis that infections cause MS is not a new one. And it was actually one of the first hypotheses proposed after the disease was defined in the mid-1800s when Pierre Marie, a famous French neurologist at the time, proposed that one day we could even have a vaccine that could prevent MS from occurring. And this hypothesis of an infective origin of MS is compelling as it could explain several aspects of MS epidemiology, such as the geographical variation of the disease in incidence in different parts of the world, the change in risk that occurs with migration, and the sudden increase in incidence in some areas where MS incidence was low or where the disease had not previously been detected. And if we look back over the past century, numerous different viruses and bacteria have been proposed to increase the risk of MS, but with conflicting results. However, in the past 30 years or so, several studies more consistently have found evidence linking the Epstein-Barr virus, EBV, to MS. And initially, this was because of the similarities between infectious mononucleosis, which is the symptomatic EBV infection, and MS. And on this slide, you can see a table from a paper from 1988, where the authors pointed out many similarities. If we look at the literature and the evidence today, not all of these hold, but some aspects, such as the gradient with latitude, is something we still see today. One of the first systematic investigations into this topic was published by members of our neuroepidemiology group and found that most infections were not associated with MS later in life. So these are data from the Nurses' Health Study. And you can see on this figure that the authors looked at measles, mumps, rubella, chickenpox, and the association was null. There was nothing there except for infectious mononucleosis. So infectious mononucleosis in this study was associated with about a twofold increased risk of MS. So a very distinct association compared to the other infections in this study. And it's interesting when you look at the studies that came after this initial study that the association was very consistent across different studies. So you can see in this forest plot that almost all the estimates falls on the right side, consistent with an increased risk of MS after infectious mononucleosis. And you can also see that the magnitude of association was quite consistent across these different studies. So it's then interesting to think about what is actually causing infectious mononucleosis. And as you can see in this figure, infectious mononucleosis, a symptomatic EBV infection, it's a lot more common during adolescence compared to earlier in life. So this shows that age at primary EBV infection is a strong risk factor for infectious mononucleosis, and that it rarely occurs when the primary EBV infection occurs in childhood. And this can explain the latitude gradient that we see with infectious mononucleosis. And with the latitude gradient, we mean that it occurs more frequently further away from the equator, and is less common closer to the equator. So on this slide, on the top figure, you can see that a much larger proportion of the population is infected by EBV in early childhood in countries like Uganda and Indonesia compared to France, Sweden, and England. And on the bottom part of the slide, you can also see that the proportion of EBV positive individuals at the time of military entry in different parts of the US also varied. And in this slide, you can see that the proportion was higher in the southern part of the US compared to the northern parts, also showing a latitude gradient within the US. So this explains the latitude gradient of infectious mononucleosis, but what can explain the latitude gradient of MS? So as you can see on this slide on the top left part, you can see that MS has a quite distinct geographical distribution around the world, that it's more common further away from the equator and less common close to the equator. And this observation has sometimes been explained by the hygiene hypothesis, which proposes that exposure to infections early in life affects the risk of certain diseases later in life because of priming to the immune system. So in this figure, you can see that on the top part of the slide, you can see the geographical distribution of MS and type 1 diabetes is quite different. It's exactly the opposite of what we see for example, tuberculosis and hepatitis B on the bottom part of the slide. And if the link between EBV and MS could be explained by the hygiene hypothesis, this has implications because if this is the case, then EBV and MS are not actually causally related, but they share the same cause. So this would mean that high hygiene could lead to a higher proportion of EBV negative individuals later in life, because these are people who escaped EBV infection in early childhood. And these individuals would then be at high risk of infectious mononucleosis because they would be infected later in life, and also at a higher risk of MS. And the reason this has implications for MS is because targeting EBV in this scenario, if this was true, would not affect MS risk. But when we look at the evidence, this is not actually what we see. And this is what we call the EBV paradox of MS. Because we see that the risk of MS in EBV negative individuals seems to be very, very low. So this is just a summary slide of different smaller studies published in 2007, showing that the odds ratio or the risk of MS in EBV negative individuals is very, very, very low. So in this meta-analysis, the odds ratio was 0.06, which is very, very low. So if we look at a better comparison of these different groups, we can look at this figure from the same paper by Dr. Scary in 2007. So on the left side, we have people who are EBV positive, but without a symptomatic EBV infection, with no infectious mononucleosis. And these individuals were likely infected in early childhood with infectious mononucleosis. So these were individuals who were infected later in life. And as you can see, EBV negative individuals have a very low risk of MS, while those with infectious mono have a high risk of MS, even if both of these groups likely experienced high hygiene or sanitation early in life. And you can also see that the comparison between the comparison risk between EBV negative and EBV positive individuals, there's a difference of a 40-fold in risk of MS. So a very drastic change in risk. So given these observations, it's hard to argue that the hygiene hypothesis can explain the link between EBV and MS. So if we summarize the evidence for a role of EBV in MS, so we know, as I've shown in the previous slide, that infectious mononucleosis has consistently been associated with increased MS risk. We also know that elevated antibody titers against EBV has been associated with an increased MS risk. Some studies also found EBV present in demyelinating lesions in MS patients, but this is a more inconsistent finding and not found in all studies. So given this level of evidence, the evidence for causality remains inconclusive. So in our study, we want to take this one step further and try to determine the role of EBV in MS. But as you can imagine, this is a very challenging task. The gold standard would be to conduct a randomized controlled trial, which is obviously not feasible. First of all, it would be very difficult to recruit EBV negative individuals, and it would be unethical to purposely expose some to an EBV infection. The alternative, and what we refer to in our paper, is an experiment of nature. So this would be to identify and follow a cohort of EBV negative individuals over time. And as we follow these individuals over time, some will be infected by EBV, others will not. And then we can evaluate the risk of MS in those who were infected with EBV compared to those who were not. And while this can seem like a simpler task, it is also a major challenge because it's very, very challenging to find individuals at risk of MS who have not already been infected with EBV. Also because MS is a relatively rare disease. So in our study, we did this, and the aim of our study was to evaluate the risk of MS after EBV infection in a cohort of individuals who were EBV negative at baseline. An advantage of doing this longitudinal investigation is that we would be able to determine the temporal sequence of events, because we know in epidemiology that an exposure must precede the outcome to be causal. So then it's relevant to ask if the EBV infection preceded the first symptom of MS, and also if it preceded the first preclinical signs of MS. In addition, we also wanted to evaluate alternative explanation for the association, to see if the association was specific for EBV, or to explore whether other viruses also played a role. If we found that the association was not specific for EBV, but rather was similar for a lot of different viruses, this could argue for a more general immune dysfunction, where some individuals are more susceptible to infections rather than an effect of EBV. So this is the abstract from our publication, where we conclude that the findings in our paper cannot be explained by any known risk factor for MS and suggest EBV as the leading cause of MS. So in the rest of this talk, we're going to discuss why we think this is the case. So while we published this paper 2 years ago, it's work that's been taking place over 20 years by the two senior authors of the publications, Dr. Alberto Asgeria and Dr. Cassandra Munger. So they started this collaboration with the US military over 20 years ago. And since then, we have documented MS cases in this cohort. So you may ask the question, why did it take so long to conduct this study? So the reason for this is related to the difficulty of identifying an EBV negative cohort of sufficient size to conduct a study of a disease like MS. And this can be illustrated by this flowchart presented here. Through the collaboration with the US military, we could access serum samples stored in the Department of Defense Serum Repository. So this biobank includes more than 60 million samples from more than 10 million individuals. Among these, we documented 955 MS cases during follow-up. So that is 955 individuals who developed MS while being on active duty and with available samples before they developed the disease. And for each case, we selected two matched controls. And among these individuals, we documented 35 individuals who were EBV negative at baseline, which means the military entry, and later developed MS, and 107 controls who were EBV negative at baseline. So thus, as you can see from this flowchart, it took a cohort of over 10 million people to end up with the 35 individuals who were initially EBV negative and later developed MS. In these EBV negative individuals, we had access to up to 3 serum samples per person, which made it possible to examine whether they were infected with EBV during follow-up. The timing of these samples is illustrated on the right side of this slide, and the left side illustrates the age at first serum sample and the age at MS onset. A strength of the study is the selection of the control, which is very commonly a weakness in epidemiological studies. So in our study, we used what we call a risk set sampling, meaning that we selected controls who were at risk of developing MS when the case they were matched to developed the disease. So this design provides an unbiased estimate of the rate ratio that would have been observed in the entire cohort of 10 million people. In addition to using risk set sampling, the controls were closely matched on sex, age, race, ethnicity, date of blood collection, and the military branch. The lab performing the serology to assess EBV status was blind to the case control status, and we arranged the samples randomly, so the sequence of cases and control samples was random within each triplet. Since this was a 20-year collaboration, the method to assess EBV status changed over time. In the first group of cases and controls, we used immunofluorescence to determine EBV status at baseline, while for the rest of the participants, we used first ELISA and then confirmed EBV status with Western blot in those who were EBV negative. So what did we find? So this figure illustrates the EBV status in MS cases over time. The green line illustrates the time they were EBV negative, the blue line when they became EBV positive, and the red dot, the MS onset. And with MS onset, we mean the first MS symptom, so the first neurological symptom of MS. And as you can see in this figure, there's a very consistent pattern for almost all the cases. They were first EBV negative at baseline. They then seroconverted, the line becomes blue, and then they became infected with EBV and then developed the first symptoms of MS. And in fact, in our study, 34 of the 35 cases who were initially EBV negative became infected with EBV before developing the first symptoms of MS. And this pattern was not the same in EBV negative controls. Only about half of them became infected with EBV during follow-up. We also included serology for the cytomegalovirus, as this is a virus that's transmitted in a similar manner as EBV, and thus is an ideal negative control. And as you can see on the right side of this slide, there was no difference in the proportion infected by CMV over time. So when comparing those who seroconverted to those who remained EBV negative, we found that EBV infection was associated with a 32-fold increased risk of MS. And being EBV negative was associated with a 26-fold increased risk. For CMV, there was no increased MS risk associated with seroconversion or being positive at baseline. So in the main analysis, we found that EBV infection was associated with a 32-fold increased risk of MS, providing compelling evidence that EBV is the leading cause of MS just because of the magnitude of the association. So in the remaining part of this talk, Marianna will discuss the rest of the results and their implications. So I'll stop sharing.
Okay, 1 second, I'll share the screen. Can you see the entire screen?
Yes.
Okay. So I'll continue now on the remaining findings and put them a bit into context. So can we really infer causality from our study, as Kjetil so nicely pointed out? Let's look at the essence of what distinguishes an observational study as ours from a randomized controlled trial, which would be the gold standard, but type of study that is not possible for an investigation of EBV and MS. What they have in common is that a finding can be a chance finding. They also have in common that some sort of misclassification of, for example, the outcome may occur, but similarly in the comparison groups. What separates the two designs ultimately is the additional risk of bias in observational studies, such as potential confounding and other biases. In the military study, we evaluated whether these additional biases could explain our findings. So the low p-value indicates that a chance finding is very unlikely. Further, we used gold standards for defining EBV serology, confirming seronegativity with 2 methods, and verified the MS diagnosis by medical record reviews according to diagnostic criteria. Selection bias and differential misclassification of the EBV status and MS diagnosis are virtually eliminated by design, as our study is a longitudinal study. Now we need to look at confounding. One concern that is often verbalized, or was often verbalized, is whether MS increases the risk of being infected by EBV. That is, can our results be explained actually by reverse causation, which is a form of confounding? We address this by conducting some additional analysis. For example, one, by measuring serum levels of neurofilament light chain, which is a sensitive biomarker of neuroaxonal injury that can capture the earliest stages of MS even before the first neurologic symptoms occur. In a previous study published by our group, we used for the first time this serum biomarker to characterize the preclinical phase of MS. And we found that MS cases started having higher NFL levels than controls 5 to 6 years before first symptom, and some even before that. EBV infection precedes the clinical onset, but does it precede the NFL elevations that occur years before? And what we found in our military study population was that serum NFL levels were not increased before EBV infection or at the time of the first EBV positive sample, but they were increased after EBV infection in MS cases, but not controls. We also saw that there was a significant within-person increase in NFL levels in the cases, but not in the controls. So from this analysis, we could conclude that EBV infection precedes increases in NFL. To further address the concern of reverse causality, we measured also antibodies titers against the human virome, and we used a method called VirScan, which is a high throughput technology for virome-wide profiling of antibodies against viral peptides. We wanted to assess whether there was some sort of immune dysregulation associated with subclinical onset of MS that could explain the stronger reactivity to EBV. Briefly about the method, VirScan uses a phage display immunoprecipitation and sequencing technology and has been validated in different populations. Here in a cartoon, you can see that a peptide library is designed using a DNA microarray that covers the full proteome of all known human pathogenic viruses. The library is then cloned into the DNA of phages that display the peptides on their surface and can be bound by antibodies in the biological sample. Antibody-bound phages are then separated by immunoprecipitation, and the phage DNA is amplified and sequenced to obtain the read counts for the antibody-bound peptides. And the peptide-specific read counts are then converted into a metric which expresses the relative enrichment of antibodies against a viral peptide. And these were the results from our VirScan analysis. This figure shows that in this figure, each dot is a peptide, a viral peptide, and the results for the cases are on the top panels, and at the bottom, you can see the results for the controls. And on the left, you can compare the pre-onset samples. As you can see, the antibody responses, if you compare the profiles, look overall very similar in cases and controls, apart from the response or antibody response to EBV, which is circled in orange. The same was the case for the post-onset samples on the right. We also examined for which peptides there was antibody enrichment. you can see that almost all peptides to which there was a significantly higher antibody binding in the cases compared to the controls mapped to EBV. These are shown in orange color here. 30 of 32 viral peptides in the pre-onset samples mapped to EBV. There was no systematic mapping to other viruses, as you can see here, that indicated a higher serological response in the cases. Finally, we also mapped the EBV peptides that showed significantly higher antibody binding in the cases to the EBV antigens. And you can see here, again, separately in the samples, that most of them mapped to EBNA1 and EBNA3, which are EBV transcription factors. So from this viral-wide screening, we could conclude that the preclinical and early clinical MS phase was not associated with an immune dysregulation that would indicate an increased susceptibility to viral infections in general. And it further underlined the specificity of the EBV-MS link. Now, can our finding be explained by other forms of confounding? There are other factors that have been consistently associated with an increased risk for MS. As you can see on this slide, family history, infectious mononucleosis, as Kjetil showed, vitamin D deficiency, smoking, and obesity in early life. They could act as confounders, also called common causes of an EBV infection and MS relationship, which would create an association, but where there is no really a causal link. To assess the potential of confounding by other factors, we calculated the E-value. And the E-value is defined as the minimum strength of association that a confounder would need to have with both the exposure and the outcome to fully explain away a specific association. The E-value in our study was 64. What that means is that any confounder would need to confer an over 60-fold increased risk of EBV seroconversion and also an over 60-fold increased risk of MS to explain the relative risk of 32 that we see in our study. None of the other factors that I mentioned in the previous slide comes close to that, and there are no other good candidates. Relative risks for these consistently associated factors range from 1.5 to 3, and the strongest risk is conveyed by the genetic risk allele HLA-DRB1 1501. To give you context, more context to an estimate of this magnitude, in Richard Dahl's study identifying smoking as a risk factor for lung cancer in the 1950s, they found a 25-fold increased risk. So we do not often see estimates of this magnitude in epidemiologic studies. In summary, based on these additional analysis, it is extremely unlikely that confounding can explain our results, which makes that our study provides the most compelling evidence to date that EBV is the leading cause of MS. Now, if EBV is the cause of MS and nearly everyone is infected with it, why do not more people get MS? And this is a question we very often get when we present the findings, or in general, people email us. And some may feel like this argues against EBV being the cause of MS in the first place, this stark contrast. But note that the fact that an exposure is highly prevalent or ubiquitous even does not stop it from being a cause of a relatively rare disease. And there are numerous examples in biology for this. One obvious example to relate back again to that most smokers do not get lung cancer. You can see here the numbers in the first study by Dahl and Hill on smoking and lung cancer. Among lung cancer patients, 99.7% smoked. Among the controls, 95.8% smoked. Just because smoking was common, it did not stop to be the cause of lung cancer, as you can nicely see here. And these numbers are actually pretty similar to the EBV prevalence in MS cases compared to controls. And also in infectious diseases, this is more the rule than the exception. Common viruses can cause rare diseases. For example, not all children infected with the poliovirus develop paralytic poliomyelitis, or herpes simplex virus type 1 only sporadically leads to fatal encephalitis. So MS can be considered a rare complication of an EBV infection. So can we prevent MS? The vast majority of MS cases could be prevented by preventing infection with EBV. So conceptually, a vaccine could prevent MS, although the development of such a vaccine remains very challenging. The other risk factors remain important as they may further modify the MS risk in EBV positive individuals, and most people are EBV positive. From attributable risk calculation, it seems like vitamin D supplementation will lead to the highest number of preventable cases. Now, what are the most pressing unresolved questions at this point? It is unclear what the mechanism is with which EBV causes MS. Very briefly about the biology, which may provide some cues, some thinking possibilities about this. EBV is transmitted through saliva and infects naive B cells that then become resting memory B cells after transition in the germinal center, in which EBV remains latent during life with restricted viral expression. But the virus retains the ability to intermittently reactivate. The resting B cells recirculate to the lymphoid organs in the oropharynx and differentiate into plasma cells, causing lytic replication cycles and shedding the virus again in the saliva. And there's a potent EBV-specific T cell response to keep the virus in check. Several mechanisms have been postulated to play a role, and popular ones are potential cross-reactivity or molecular mimicry to EBV and central nervous system antigens. Another one is that EBV could modify the immune response. EBV is, as you know, an oncogenic virus and transforms B cells. Could it have the potential to transform B cells in a way that causes MS? Regarding molecular mimicry, a Nature paper came out briefly after our paper came out, reporting a high affinity molecular mimicry between the EBV transcription factor EBNA1 and the central nervous system protein glial-CAM, which is a glial cell adhesion molecule. It will be interesting to see whether this finding can be reproduced. A related question is whether EBV is only a trigger or also a driver of the disease. If EBV is a trigger for MS, the virus causes a break of immunological self-tolerance and starts a self-perpetuating autoimmunity cycle that leads to tissue damage. If EBV is also a driver for the disease, the disease processes may be driven by anti-EBV immunity, and autoimmunity may arise as part of this anti-EBV immunity, but would cease if infection with EBV is eliminated. And the answer to this question will also define the most promising next steps and therapeutic strategies. They could range from a vaccine to immunotherapy or antivirals. One of the lower hanging fruits is to test antivirals as a treatment in MS patients. A drug used to treat HIV tenofovir alafenamide has recently been shown to have potent anti-EBV properties. And we have also identified MS patients treated with this drug that seemingly have a very positive response on disease activity. And it is exciting that we have been recently funded by the EU to start a trial to test this drug in MS patients. Oh, yeah. Okay. And to summarize the whole talk, the evidence supporting a role of EBV in MS etiology has been converging over the past decades. A recently published longitudinal study in the US military cohort following EBV negative individuals for EBV seroconversion and development of MS provides the most compelling evidence to date that EBV is the leading cause for MS. Understanding the mechanisms underlying the EBV-MS link may help us lay the groundwork for a cure and primary prevention. And I would like to acknowledge the senior investigators and driving forces on this project, Dr. Skeri and Dr. Munger, which started the collaboration with the US military 20 years ago and are an example of endurance. And I would also like to thank all the wonderful collaborators from the different institutions that made this project possible and acknowledge our funders. And thank you for your attention.
Thank you both so much. That was a fantastic talk. I'm going to start off by bringing up some of the questions that were posted to the chat for you both to address. And then I have many questions myself, but I'll let other people jump in first. So one of the questions is from Anders. Could it just indicate an infection-shielded upbringing? So this is likely related to the hygiene hypothesis you were discussing.
So, I don't think the findings can be explained by an infection-shielded upbringing. I think the reason why some people remain EBV negative could be related to hygiene, but the hygiene cannot explain the link between EBV and MS, would be my answer.
And then Anders also mentioned that they published about 30 years ago about an increased risk of MS if you had mononucleosis. So similar to what you both have been echoing, this has been a really intense area of focus for a long time. And it's fantastic to see the data really pulling together and just the scope to have 10 million samples that come down to 35. That's impressive. Andrews also asked if entering the military being EBV negative and CMV negative, one would think it would be more likely to get infected with EBV than CMV. And I think I would agree. That seems like, I don't know if you each have thoughts.
Yeah, no, so absolutely. But I think also still, because EBV is more prevalent than CMV, but I think still what's interesting to see afterward is also the timing of infection, because to get this 32-fold increased risk, like if EBV was not associated with MS risk, you would see that sometimes the infection would fall after MS onset, sometimes before, like there would be sort of like a normal distribution of the timing. So I think also the timing aspect that almost like all of them got before the first symptoms and also before NFL elevation argues for that it's not just happening as it would at the same rate as in controls.
Sure. We have another question from Raymond. Any EBV fingerprints or viral DNA in neurons from cadavers from MS patients? That's a really interesting question. I would love to know too.
I don't know. Marianna, do you
want to?
Yeah,
there have been studies that identified EBV in brain postmortem MS patient brains, but it wasn't a consistent fact. Other groups couldn't replicate the findings. So there has been a discussion whether, for example, EBV needs to infect the central nervous system or not. But it could also be a different mechanisms through the immune response to EBV, which happens in the periphery. So this is not a proof that EBV cannot cause MS, I would say.
And we have another question or comment that they're having indication of EBV reactivation, similar to other viruses like herpes. So what is your take on that, if that's a contributing factor to MS?
Lytic replication? Mm-hmm. Yeah, that's a very interesting question. Obviously, we don't know the mechanisms. We can speculate. It's interesting to see that the antiviral that we're going to test is actually targets the DNA polymerase. So it seems to point towards a more lytic replication mechanism. If we see an effect in the trials, then it would show in that direction. That's why testing an antiviral that is safe in MS patients would be the lowest hanging fruit, because it could reversely show or indicate the mechanism.
I think that's a really intriguing approach. I'm curious, for the design of that, how long are the patients being treated with the antiviral?
That's another excellent question. We have been discussing the design of such a trial for years. It's very, very challenging today to conduct clinical trials in MS patients, because since the 90s, many drugs have been developed that are disease modifying. And today we have very potent disease modifying drugs that stop the neuroinflammation and the relapses. But there's less clear evidence, and it doesn't seem to stop the disability progression. So conducting such a trial is really challenging. And even during our funding period now, when we had a design and a protocol, and now we're even again discussing to modify that design because new evidence came out. And what is the correct comparator? We don't want to use a comparator where patients are treated with B-cell depleting therapies that then, if we don't see anything, we don't know
right.
why. So it's very challenging, and also the ethical aspect that we cannot keep people on an antiviral when we don't know yet if it's effective, because we know that early treatment in MS is very important for the long-term outcome.
Yeah, certainly a complicated issue. Does anyone want to speak up about a question?
Sorry?
Oh, I was just opening it up. I have many questions, but I was going to let people unmute themselves and ask if they would like.
I see there's another question in the chat, I think, of the Q&A.
Yes. Is the MS risk related to IM or to the delayed age of primary infection, even if asymptomatic?
So it's hard to separate the two factors, but I think it doesn't seem like infectious mononucleosis is required to develop MS. So the EBV infection would be the underlying factor, and then the risk is further enhanced if you have a symptomatic infection. But then again, since infectious mononucleosis is so closely related to age at primary infection, it's hard to distinguish the two.
Yeah, it's certainly a complex interplay between these different phenotypes. What is your thoughts on, I thought the data you had for the biomarker on neurofilament was really intriguing, that it was detectable up to 5 years before disease onset. So is that an FDA approved test? Is that something that people could be tested for if they have a hereditary risk for MS, just to be aware that that's a potential down the line?
Yeah.
So this is the NFL.
NFL, yeah, it's a good point. People are trying to explore the clinical use of neurofilament light chain, and whether we could help to detect individuals earlier, or it could be used for clinical staging, in addition to MRIs, for example, or symptoms. But it's a little bit challenging because NFL elevations are unspecific. There are other neurodegenerative conditions that can lead to a higher NFL. But in MS, I would say what is in favor of a use is also the age at onset, is a younger age at onset. And maybe, as you say, if we further stratify people at risk with family members of MS, there is no such test that you can buy in the pharmacy and conduct yourself.
No, yeah.
This is something you would discuss with your medical provider, but it's not a standard clinical practice to measure NFL at this moment, but there's a lot of research ongoing.
No, it's certainly an exciting avenue that hopefully we'll get to hear a lot more about. I see Alfredo posted a question. Again, great talk. Did I see correctly that in the ViraScan approach, there was no antibody signature for HHV6, which is another very common virus contracted early on in life and very common in the population?
Yes. So no, there was no signal for HHV6. And I know there are studies that have compared HHV6A and B. We did not find any systematic differences for this virus.
Wow, that's quite interesting. Yeah, and so I guess one other question I have is if, in particular, you had recommended vitamin D is the main takeaway at this point. And then, oh, I see Sten posted a comment on if immunotherapy treatments for MS, if those are being utilized and what those are.
You want to?
I mean, you can go ahead if you want.
There are different avenues. I'm not an expert, but different avenues are being tested. For example, one which unfortunately showed negative results was a study run by Atara as T-cell therapy. So the idea that the T-cell response in MS patients may be defective, and we could treat with immune competent T cells, but unfortunately the results were discouraging in the last trial. People are exploring different mechanisms. I think it will be a hand-in-hand thing, mechanistic studies, trials. Hopefully we'll get closer to the answer.
Yeah, absolutely. I mean, I think it's clear that there's a lot of interesting, valuable questions to pursue in this line of research. And I'm very grateful to you both for speaking today. And this was quite a fantastic talk. And we appreciate you joining us.
Thank you.
Thank you for having us.
Thank you. And if there are no further questions, then I'll let everyone go. And I look forward to seeing you at the next webinar.
Thank you.
Thank you both so much.